How do you investigate the effect of light intensity on the rate of photosynthesis? Hi, my name's Dr. De Bruin. I'm a GCSE and A-level science teacher, and I also make revision videos on YouTube. If we're going to measure the impact of light intensity, then we need to be able to change light intensity. So the easiest way to do that is you have a lamp, you have a plant, and you have a ruler that measures the distance between them. And as you move the light closer, then the light intensity increases. And if you're taking higher tier, you could even talk about inverse square law. So if you double the distance, then you quarter the amount of light that's being received. Now we can't just use any old plant because we want to be able to measure the rate. And for that we need to know what products are being produced. So if we use an aquatic plant, something like LED, then it photosynthesizes underwater and it releases bubbles of oxygen. And you could count those bubbles or you could collect the gas and measure the volume. Now in order to work out the rate, we need to know how fast those bubbles are being produced. So you're going to need to count bubbles per minute or measure the gas produced per minute. So that's the basic experiment. Now let's talk about all the little details. Number one, before you start measuring the bubbles that are produced by this plant, you're going to need to leave it with that light to kind of acclimatise for a bit. It's not just going to suddenly start photosynthesising like mad just because you gave it some light. Number two, if that light is too close, it could heat up the water and that would also affect the rate of photosynthesis. Then our experiment wouldn't be valid. So your two best options for this are to either use an LED lamp and explain that you're doing that because LEDs don't get hot, or to use a heat screen. The rate of photosynthesis will also be affected if the plant is limited by a lack of carbon dioxide. So rather than keeping our aquatic plant in a beaker of water, we're going to use sodium carbonate solution, because when that sodium carbonate dissolves, it can break apart to produce carbon dioxide. To check that this experiment is repeatable, you're going to do each light intensity more than once so that you can compare the results. This allows you to remove any anomalies and also calculate a mean for each light intensity. And once you've got all of your volumes or all of your numbers of bubbles, then you're going to need to actually calculate the rate of photosynthesis. This could be bubbles per minute or centimetres cubed of oxygen produced per minute. Also, just double check the question, because sometimes they ask you a very similar method question, but not about the required practical. So say if they've said investigate the different colours of light, then you need to talk about how you would change the colour of the light. And the best way to do that would be to put a coloured filter in front of the light. Because even though you can buy different coloured light bulbs, they might have different intensity. And the whole point is that you only want to be changing one.